Welcome back, everyone, to another deep dive into today's most exciting genomics research. 5 new papers, all hot off the presses, are waiting for us.
Let's dive right in.
Today's lineup has it all. We've got microbial communities, some very interesting work on date palms, and even a new way to think about how we analyze individual cells, all thanks to the power of genomics.
Incredible how much this field is revealing about the world around us.
Absolutely. So first up, let's talk about metagenomics. This new paper introduces something called CoverM, a new software designed to help us analyze those bustling cities of microbes we call microbial communities. Like the ones living in our gut or out in the soil.
Everywhere, really. And what's cool about CoverM is that it takes what we call a genome-centric approach.
It
It goes beyond just identifying the species present. Instead, it focuses on the complete genetic makeup of the whole community.
like comparing an entire ecosystem to a single species.
Precisely.
The article mentions this thing called Mohs depth arrays. Sounds complicated. What's that all about?
Well, it's a bit technical, but essentially, CoverM uses these arrays to efficiently figure out how much of each genome in a sample is represented in the sequencing data.
Coverage.
Yes, exactly. Coverage basically tells us how complete our picture of the microbial community is.
So higher coverage means a clearer picture.
Exactly. Think of it like having a higher resolution image, giving us a more accurate and reliable view of the community. What's also exciting is that CoverM is free and open source. It even has Python and Julia interfaces, making it incredibly accessible to researchers.
That's great news. Imagine all the applications. Personalized medicine, environmental monitoring. Amazing. All right, let's switch gears a bit from the microscopic world to something, well, a bit more delicious. This next study used genomics to uncover some secrets about date palms.
Date palms.
Date palms. Specifically, how to get bigger, better dates. Commercially, dates are harvested at a stage called the tamar stage. This study really focused on what genetic factors influence fruit size at this specific stage.
Yeah.
Thinking about improving yields, market value, that kind of thing.
Important stuff, for sure.
So they used GWAs to pinpoint the specific genetic markers linked to date size. Can you walk us through that?
GWA stands for Genome-Wide Association Study. It's a really powerful method that helps us connect specific genes to certain traits by comparing the genomes of dates with different sizes.
You can find the regions of DNA responsible for those differences.
Exactly. Like finding the genetic recipe for a larger date.
Fascinating. What genes did they actually discover? What's behind those jumbo dates?
They found a few, and they're involved in all sorts of fundamental processes like cell growth, differentiation, and even those complex signaling pathways that regulate these activities. So basically, they've uncovered part of the genetic blueprint that determines how a date palm grows its fruit.
So theoretically, this knowledge could be used to breed dates with very specific traits. Size, sweetness, maybe even color.
Absolutely. Imagine a world with a greater variety of dates. More delicious, more nutritious. This research really opens up some exciting doors for targeted breeding programs.
A world full of delicious dates. Sounds pretty good to me. All right, let's move on to our next paper. This one takes us deep into the world of single-cell RNA sequencing, or cRNA-seq for short.
This is getting down to the individual cell level.
Right. This study dives into the complex world of cell clustering using cRNA-seq data. Why is this kind of clustering so important?
Well, it's crucial for understanding the incredible diversity of cell types in our bodies. Imagine organizing a city into neighborhoods based on, let's say, the activities and characteristics of its residents. That's kind of what we're doing here.
Grouping cells with similar gene expression profiles.
Yes, to identify different cell populations and their unique functions.
This paper proposes a new approach using something called hypergraphs. Sounds a little science fiction-y. What are they exactly?
Hypergraphs offer a more, I guess you could say, nuanced way to represent the relationships between cells. Think of a social network where people form groups based on shared interests. Traditional methods only capture those pairwise connections, but hypergraphs can represent groups of any size, even those complex multi-way relationships between them.
So instead of just looking at one-to-one interactions, you're seeing how cells are connected in a much broader context.
Exactly. This is especially important when you're dealing with complex biological systems where cells are constantly interacting and influencing each other.
This study developed 2 specific methods using this approach, DIPHW and CoverM's. I'm not sure I can say those 10 times fast. For those of us who aren't genomics experts, what's the main takeaway here?
Simply put, these methods offer much better accuracy when clustering cells, especially with complex data sets. Traditional methods might miss those subtle but crucial differences between cell types, but these new methods can handle that complexity.
It's like upgrading your microscope to see cells with much greater detail and clarity.
It really is groundbreaking work.
Our next study sticks with CRNA-seq, but with a fascinating twist. Time.
Time series, CRNA-seq, powerful stuff.
It allows us to see how a cell's gene expression actually changes throughout its life.
It's like watching a movie of a cell's life, capturing all the dynamic changes as it grows, divides, and performs its functions.
That's a great way to visualize it. But I imagine there are challenges in this type of analysis.
Oh, absolutely. One of the biggest is ensuring accurate timestamps for every single data point. If those timestamps are off, you can end up with a jumbled mess, like a movie with all the scenes out of order.
You wouldn't be able to follow the plot.
Exactly. Inaccurate timestamps, or what we call noisy timestamps, can really mislead us about a cell's developmental journey.
So how do we make sure those timestamps are on point?
That's where this new study comes in. They've introduced a new timestamp calibration model called SkiPace. Think of it as the film editor for our cellular movie.
Ensuring all the scenes are in order.
Precisely. It cleverly identifies and corrects those noisy timestamps, making our analyses much more reliable and insightful.
Sounds like a real game changer. Did they actually test this SkiPace model?
Oh, yeah. They tested it on both simulated and real data sets.
And
The results were great. SkiPace significantly improved the performance of pseudotime analysis, which is a technique for ordering cells along their developmental path.
making the cellular movie much clearer.
Exactly.
It's incredible to see this constant innovation happening in genomics research.
It really is.
And speaking of pushing boundaries, our last study for today dives into one of the fundamental challenges in genomics: assembling complete genomes.
A massive task.
Imagine trying to put together a giant jigsaw puzzle. Now imagine that puzzle has billions of pieces, many of which look almost identical.
Now you're getting the picture.
This study actually gets into the nitty-gritty details of how much sequencing data or coverage and what read length you actually need for a successful assembly, especially for a diploid genome. Can you remind us what those terms mean?
Coverage refers to how many times each part of the genome is actually sequenced. More coverage means more pieces of the puzzle, so to speak, which generally leads to a better assembly. Read length, on the other hand, refers to the size of each individual piece of sequencing data. Longer reads are like having bigger puzzle pieces, making it easier to put them together.
It's all about finding the right balance between the amount of data and the size of each piece.
Exactly.
This study really dives into the theoretical limits of what's possible, like what's the minimum information needed to put together a whole genome. They also looked at how different assembly algorithms, those computer programs that piece the genome puzzles together, actually perform in practice.
Essential for making this whole process more efficient.
What did they learn from all that?
Well, one interesting finding was that many existing algorithms actually need higher coverage than the theoretical minimum to do a good job. And one of the reasons for that is the presence of something called double repeats in genomes.
Double repeats.
Imagine having two identical puzzle pieces. It's tough to figure out where they go without some extra information. That's what these repeats do to the algorithms.
They throw a wrench in the works.
Exactly. The research highlights the need for better algorithms that can handle these repetitive regions without breaking a sweat. Longer read lengths can also help by providing more context for placing those tricky repeat sequences.
So better data and smarter algorithms. That's the key to more accurate genome assembly in the future.
Definitely. And this research is laying the groundwork for making that happen. And this research has big implications too. Think personalized medicine, evolutionary biology, even conservation efforts.
It's amazing how such a complex task can have such real-world impacts. It really highlights the importance of basic research, pushing the boundaries of what we know. Speaking of pushing boundaries, let's circle back to a study we talked about earlier, the one that looked at cell clustering using layering cRNA-seq data.
Ah, yes. Getting down to the individual cell level.
Exactly. This technique lets us group cells based on their gene expression profiles. We're essentially identifying different cell types and their unique functions.
Right, and we talked about how traditional methods for cell clustering often miss the bigger picture. They rely on those pairwise comparisons, which it can be limiting when you're dealing with complex systems.
That's where hypergraphs come in.
Exactly. Hypergraphs help us capture the full complexity of those cellular interactions.
Can you remind us how they differ from the traditional approaches?
Imagine you're mapping out a social network. Traditional methods would only show who's directly connected to whom. But with hypergraphs, you can actually represent groups of people with shared interests, even if they aren't directly linked. It's a more nuanced way to capture the interconnectedness of the network.
So applying this concept to cells, hypergraphs can reveal those intricate multi-way relationships that would be missed by simpler methods.
Precisely. Instead of just one-to-one interactions, we're seeing the full spectrum of how cells relate to each other. It gives us a much richer understanding of how cells function as a collective.
The researchers in this study developed two specific methods using hypergraphs, DIPHW and CoverM's. What should our listeners take away from this development?
The key takeaway is that these methods offer significantly improved accuracy when it comes to clustering cells. And this improvement is especially pronounced in complex datasets, where traditional methods might miss those subtle but crucial differences between cell types. This is particularly important when studying diseases like cancer, where even small differences can have big implications for treatment.
So it's not just about identifying different cell types, it's also about understanding the nuances of how they relate to each other. This could have huge implications for personalized medicine and drug development.
Absolutely.
Now, let's shift gears again and revisit another study we touched on earlier, the one that explored the world of time series cRNA-seq, the one that lets us track how a cell's gene expression changes over its lifetime.
Like watching a cell's life story unfold, seeing all the dynamic changes as it grows, divides, and performs its functions.
It sounds incredible, but I imagine there are technical hurdles to overcome when analyzing this type of data.
For sure. One major challenge is ensuring that the timestamps for each data point are accurate. Think of it like watching a movie where the scenes are all out of order.
It would be pretty confusing.
Exactly. Inaccurate timestamps can lead to misleading conclusions about a cell's developmental trajectory.
So how do researchers ensure those timestamps are accurate?
This study introduced a new timestamp calibration model called SKES. It's like having a film editor for your cellular movie.
Making sure all the scenes are in order.
Precisely. It intelligently identifies and corrects those noisy timestamps, leading to much more reliable and insightful analyses.
So SketchBase acts like a quality control step, ensuring that the story we're seeing unfold is an accurate representation of the cell's life.
Yes. It ensures that our understanding of cellular dynamics is based on reliable data.
Accuracy seems to be a recurring theme in many of these studies, whether it's getting the right coverage in metagenomics, pinpointing those genetic markers in GWS, or correcting timestamps in time series cRNA-seq.
It's about making sure we're getting the most out of the data.
Let's talk about one final study that highlights the importance of accuracy. This one focused on the challenge of assembling complete genomes.
A computationally demanding task, for sure.
It really is like piecing together a giant jigsaw puzzle with billions of pieces.
And many of those pieces look almost identical.
This study delved into the theoretical limits of genome assembly and the performance of different algorithms. Can you give us a recap of what they found?
One key finding was that existing algorithms often require higher coverage than theoretically needed to achieve a good assembly. And this is partly because of those double repeats in genomes.
Those pesky double repeats.
They confuse the algorithms.
What can be done to overcome this challenge?
The study suggests that longer read lengths can help. It provides more context. They also emphasize the need for better algorithms, ones that can handle these repetitive regions more efficiently.
So better data and smarter algorithms. That's what we need to achieve more accurate and complete genome assemblies.
Precisely.
Why is this so important? What's at stake here?
Well, accurate genome assembly is crucial for a wide range of applications. Think personalized medicine, evolutionary biology, even conservation efforts. Imagine trying to understand a complex machine without a complete blueprint.
It would be nearly impossible.
And that's essentially what we're doing when we study an organism without a fully assembled genome.
It's incredible to think how something as seemingly abstract as genome assembly can have such tangible impacts on our world. It really underscores the importance of investing in basic research and constantly pushing those boundaries. As we conclude this deep dive into the latest genomics research, what are your final thoughts?
What strikes me is the incredible pace of innovation in this field. Researchers are constantly developing new tools and techniques to analyze this huge amount of data that sequencing technologies generate. It's an exciting time to be in genomics, and I'm eager to see what the future holds.
I completely agree. It's clear that genomics is revolutionizing our understanding of life, and it's only a matter of time before these discoveries translate into real benefits. Benefits for human health, agriculture, the environment. Before we wrap up, I want to remind our listeners that science is a collaborative process, and we always encourage you to share your thoughts and questions. What mysteries of life are you most curious about? What scientific advancements are you most excited to see in the future? Let us know.
And it's not just human health. Agriculture is benefiting tremendously from genomics research, too. Think about those date palms we discussed earlier.
Right. Using GWI to identify genetic markers linked to fruit size. Imagine the possibilities for improving crop yields and creating new varieties with desirable traits.
Exactly. And it's not just dates. We're seeing similar advancements in all sorts of crops. From disease resistance to drought tolerance, genomics is playing a key role in creating a more sustainable and food-secure future.
It's really amazing to see how these discoveries are being applied to address real-world challenges. We've talked about analyzing entire genomes and understanding how genes are expressed. But what about zooming in on the individual cell level? We touched on this earlier with single-cell RNA sequencing.
Ah, yes, the cRNA-seq. It's revolutionized the way we study cells.
One of the studies we discussed highlighted a new approach to cell clustering using ccRNA-seq data. Can you remind us why this is so important?
Well, it all comes down to understanding cellular diversity. Our bodies are made up of trillions of cells, and each cell type has its own unique function. By grouping cells with similar gene expression patterns, we can get a much clearer picture of how different cell types contribute to overall health and disease.
The researchers in this study use something called hypergraphs to represent those complex relationships between cells. Why are hypergraphs so crucial for this type of analysis?
Traditional methods often rely on those pairwise comparisons, which can be limiting. Hypergraphs, on the other hand, allow us to capture those multi-way interactions that are so crucial for understanding how cells function as a collective. It's like moving from a two-dimensional map to a three-dimensional model. You get a much richer and more accurate representation of the cellular landscape.
And this increased accuracy has some pretty significant implications, right?
Oh, absolutely. Especially for fields like personalized medicine and drug development. Imagine being able to pinpoint the precise cellular changes that occurred during disease progression, or even identify new drug targets based on a more nuanced understanding of those cellular interactions.
The possibilities are truly exciting. We've also discussed how time series cRNA-seq lets us track changes in gene expression over a cell's lifetime. But accurately capturing those changes relies on having precise timestamps for each data point.
Time is of the essence.
The study we discussed earlier introduced a new method called SyncBase for calibrating those timestamps. Why is this such an important step?
Well, think of it like this. If you're watching a movie and the scenes are out of order, it's pretty hard to follow the story, right?
It would make for a pretty confusing movie.
The same principle applies to time series cRNA-seq. Those timestamps are crucial for understanding the dynamics of cellular processes. Without them, we're essentially missing key parts of the story.
So SyncBase ensures that we're getting the full picture.
Exactly. By correcting those noisy timestamps, we can be much more confident in our analysis and interpretation of the data.
It's incredible to see how genomics research is constantly pushing the boundaries of what we know. From deciphering the secrets of our DNA to unraveling the complexities of cellular life, it's clear that this field is having a profound impact on our understanding of the world around us.
I couldn't agree more. And what's even more exciting is that we're really just at the beginning of this journey. There's still so much to discover.
That's what makes it so fascinating. As we conclude this deep dive into the latest genomics research, what key takeaways do you hope our listeners will remember?
Well, first and foremost, I hope they come away with a sense of awe and wonder at the incredible complexity of life at the molecular level.
It really is amazing.
And I also hope they appreciate the incredible ingenuity and hard work of the researchers who are pushing the boundaries of what's possible in this field.
Absolutely. It's inspiring to see the creativity and dedication that goes into these discoveries.
And finally, I hope our listeners understand the profound implications of this research. From personalized medicine to sustainable agriculture to conservation efforts, genomics has the potential to address some of the most pressing challenges facing our world today.
It's an incredibly exciting time to be following this field. We've covered a lot of ground today, but there's always more to explore. We encourage our listeners to stay curious, keep learning, and never stop asking questions.
And we'd love to hear from you. What mysteries of life are you most curious about? What scientific advancements are you most excited to see in the future?
Thank you for joining us on this deep dive into the fascinating world of genomics. Until next time, keep those minds hungry for knowledge.